Section 18 in Making Sense of Statistics, a conceptual overview, will be discussing sample size. So, in previous sections we've discussed the importance of using random sampling and random samples whenever possible. Remember, using random sampling, we are then, as researchers, able to draw unbiased sampling. However, also an unbiased random sample still contains sampling errors. So basically, one of the things is that sampling errors can be evaluated with inferential statistics. But inferential statistics cannot be used to evaluate the role of bias, which is why in the first place we are trying to eliminate bias by using random sampling. So sometimes this really confuses students. Sometimes by luck of the draw, we may actually... Now, as a general rule, we can say the larger the random sample, the smaller the sampling error. So what we mean by that is that the larger the random sample is, the more precise the results are. When we're talking about precision, statisticians define precision as the extent to which the same results would be obtained if another random sample were drawn from the same population. So the basic way to increase precision is to increase your sample size. Now that is kind of what we said. Now, while we might look at it from that perspective of if it's always better to have a larger sample. Well, not necessarily. So now let's talk about how increasing sample size also produces diminishing returns. So while increasing the sample size increases precision, we actually, increasing sample size may produce diminishing return. And this is less obvious. So one of the things that we'll want to look at now are examples that clarify this statement. So let's say that we have added 10 individuals to our sample. So we've collected our data, and let's say, so now we've had in example 2. So let's say that each of these persons represents 100 people. So researcher B in this example we have 1,000 persons that we have in our sample, which is a large sample. Going to add 10 persons. So you see that we have the 1,000 people that are represented at the top, and now we add 10 more persons. So we've only increased our sample size by 1%. Now our sample size is 1,010 by selecting these 10 individuals. And what this does is when we're looking at this from a different perspective, we see that the researcher in example 1 could have a substantial change in results by increasing the sample size from 10 to 20. But in contrast, the researcher in example 2, and the one that you see now, can really expect little effort on the results by adding 10 to a sample of 1,000. Well, national opinion polls usually have 15 samples of 1,500, and these are usually adequate. But adding more people is basically diminishing return. It means, you may not be able to have more precision just by adding more people. And one of the downsides to adding more people to the sample is cost. So, we can discuss it also from a level of it might actually cost you more to add 10 or 100 more people when what you have is sufficient and adequate. So when we're talking about national opinion polls, to do 5,000, you're not going to have as realistically any more precision than you would at 1,500, based on workload for the researcher. Time is money in research. So it's not cost effective, and that's something that a lot of times we have to view. Adding more individuals to the sample really produces very little increase in the precision of the result. So now, one thing that we can do and can look at is that we explore differences among groups of individuals. And so the smaller the anticipated difference in the population, the larger the sample size should be. So for example, let's say a new drug has been developed for relieving headaches, for migraines. And so the pharmaceutical researchers believe that the new drug will only be slightly more effective than the older or the existing drugs. And so with very small samples, so one sample taking the new drug and one sample taking the existing drug, researchers may not be able to detect a small difference because the results will be very imprecise to the limited sample size. However, let's also look, though, that small samples can identify a very large difference. So let's say we're testing, as researchers, a new antiviral treatment for a new virus. And we use random samples of 100 individuals for the experimental group and 100 for the control group. And all individuals in both groups were sick with the virus. So let's suppose that those who received the new treatment, this experimental group, the new treatment, 5 died, which out of 100 would be 5%. While those who did not receive the new treatment, they had the control group, 45 died. Now, because of the large difference, this should be actually thought about as a very promising result, even though we studied only 200 individuals. So let me put this in perspective, and let's think about this from the practicality of this. How many of you would decide to take the experimental drug if you became sick with this virus? And if you decided, to take the treatment, you are acknowledging the importance of the large difference in a relatively small sample. Basically, what you are acknowledging is the difference of persons not taking the experimental drug. More of them died. So you are acknowledging the importance of a large difference obtained with 2 relatively small samples. Would you reject the treatment? Would you wait until the results were later replicated in studies with larger samples? Most of us, when looking at the fact of facing death, would probably go with the experimental drug regardless at this point, right? Because you only had 5% of the persons in the experimental group died, while those in the control group, you had 45% of them die, and so that would be 45 persons. You would more likely die in the control group than the experimental group. So, with populations with very limited variability, and a lot of times when we did this, we look at it from the standpoint of twins. You know, Small samples can yield precise results. So what we can say is if a anthropologist discovers a lost tribe that consists of 1,000 identical twins, because there's no variation, even if they're raised identically, then studying one or a sample of one out of 1,000 should actually yield very precise results. So, think about that when we think about sample size with limited variability. So small samples can yield precise results. If you look at this through some of the previous studies, you could look at it from groups of Amish where we have small groups of Amish persons who are isolated, and we are able to study a very small group. Perhaps the nun study is another good example. We might have very limited variation. Now let's also look at this from the more variable the population, the larger the sample size should be. So let's say we want to estimate the math achievement of 6th graders in a large metropolitan school district, and we draw a random sample of 30 students. Because there is likely to be tremendous variation in math ability in a large district, a sample of 30, 40, 50, or 60, since it's random, might have very misleading results. You might have many high achievers or honor students. So using a much larger sample would give you better results. Now, when studying rare phenomenon, you actually should obtain a large sample. So this is another principle in determining sample size. When studying a rare phenomenon, you should use a large sample. In fact, large samples are usually required. So an example would be HIV in college students. If we are looking at this, we have in 2010, we had new HIV status in 13 to 24-year-olds of around 12,200 new cases. We also had 10 million new STI cases in 2010, age 15 to 24. So, let's look at this from a standpoint. If we have 100-person sample, we might not find any students with HIV. And so what we might assume is that no college students have HIV. Especially if we're wanting to estimate the percentage of college students who are HIV positive. Now, we would probably find no cases because the disease is relatively rare and it is unlikely to be evident in this small sample. So we might conclude erroneously, mistakenly, that no college students are HIV positive, but by using a sample of thousands, we probably would be able to gain a more precise estimate of the small percentage who are positive. Now, what if we studied STIs? Because I just told you that there were 10 million new STI cases in 2010 for ages 15 to 24. We probably, within a 100-person sample, would be able to actually find persons who had an STI, just perhaps not HIV. So when using a large sample, remember, does not correct for bias. If you ask a group of people. In fact, if we were within the classroom, I could ask you a question about a topic, and it could be anything from how do you feel about homelessness, how do you feel about underage drinking, how do you feel about abortion, how do you feel about legalization of marijuana. Currently we're in an election year. We would have a variety of answers on who persons would want to vote for. And also, we think about this from a standpoint of, when we talk about, let's say we're in college and we're discussing free college. I would dare say that if I'm in a college classroom and I ask a question of how many persons in this classroom believe that everyone should receive a free college education, I am pretty sure that all of you in the classroom would go, absolutely. Why? Because we're paying for college. We think it should be free. However, if we ask a different group of people, persons who are not in college, persons who believe that trade school is the way to go, persons that believe that apprenticeship is the way to go, you might not get the same answer. So, it doesn't always correct for bias just to have a large sample size. Now, of course, the more varied the population, the larger our sample size should be. So here again, the more varied the population, you should always use a larger sample. However, in general, it is still better to use a small unbiased sample than a large biased sample. Using random process to select participants eliminates bias. So one of the things that we also have to remember is determining sample size is a complex process. There is no simple answer to the question of how large a sample should be. Sample size will be discussed in future lectures. Again, and within the various contexts, because sample size affects the results obtained with inferential statistics, which is now what we're moving toward within our lecture series. And this concludes section 18, sample size in making sense of statistics.